Hello, my name is David Bisaro, and I'm a professor in the Department of Molecular Genetics at The Ohio State University. And it's a pleasure for me to talk with you today. I've spent most of my career in working on the DNA-containing geminiviruses, and in fact, we still are very active in that area. But today's talk is about viroids. And before I begin, I would like to acknowledge two individuals. First, my late colleague, Dr. Biao, who was an outstanding scholar and investigator, and who is responsible for me getting interested in viroids in the first place. And I'd also like to acknowledge Dr. John Wu, a graduate student and postdoc in the lab, who single-handedly is responsible for all the data I'm going to show you today, and much more besides. So the title of my talk is really quite specific. It's about biased Pol II fidelity in contributing to conservation in the potato spindle tuber viroid genome. But before I go into that, I'm going to give you some background about viroids. Some of you may not be familiar with them. So viroids are the smallest known plant pathogens. They are circular single-stranded RNAs ranging in size from about 250 to 400 nucleotides. They encode no proteins, and they're basically unencapsulated infectious RNAs. And these were discovered in 1971 by Ted Diener, who's shown here up on the left. Now, viroids are placed in two families, depending on their structure and functional characteristics. Avocado sunblotch viroid is the type member of the absent viroidae. These are mostly branched structures, and they replicate in the chloroplast of infected plant cells, and they have intrinsic ribozyme activity embedded within the RNA. This talk is going to be concerned with the PSTVd viroidae, specifically the type member potato spindle tumor viroid. These are mostly rod-shaped, and they replicate in the nucleus and do not have ribozyme activity. Now, viroids have to do a lot of things, as viruses do, and they accomplish all this simply using RNA sequence and RNA structure, a really remarkable thing. They have a lot in common. Once they get into the cell, they need to find the appropriate organelle. In the case of some, the chloroplast. In the case of PSTVd, the nucleus. They replicate within the organelle. They are then exported from that organelle and go to adjacent cells. So they must traffic cell to cell and eventually make their way into the vascular tissue, specifically usually the phloem, where they can spread long distance relatively rapidly and exit the phloem to invade new cells. And while doing all this, they need to evade host defenses. So there's really a lot to do for small RNA. Now, a little more about PSTVd. Its genome is shown here. It's a circular non-coding RNA, as I mentioned before, of 359 nucleotides. And again, all functions required for infection are mediated by the RNA sequence and structure itself. Now, the genome folds into a rod-shaped structure, so it's very highly structured, that contains 17 GU base pairs. These are wobble bases mostly. And these are indicated by the red ticks at the bottom here. And 27 loops and bulges, which are numbered across the top here. Now, these appear to be non-paired in this diagram, but in fact, that's not the case. These all have 3D structure due to non-Watson-Crick base pairing, base stacking, and other sorts of interactions. For example, a model of the loop 27 structure is shown here below. Now, we've been greatly aided in our research by two individuals I want to acknowledge, Neocles Leontis and Craig Zerbel, both at Bowling Green State University. Unfortunately, Dr. Leontis passed away very recently, but without the input of these outstanding structural biologists, much of what we've done so far would not be possible. A focus in the lab is to determine the roles of the 3D structured loops and GU pairs in PSTVd trafficking and replication. So let's talk about that for a minute. So like plant viruses, viroids move cell to cell through structures called plasmodesmata, which are cytoplasmic connections between cells that actually bridge the cellulose cell wall. And ultimately, of course, they get into the vascular system, as I mentioned earlier. Now, as infectious non-coding RNAs, viroids are really excellent models to study RNA transport in plants. And what we've learned so far in many years of investigation in Dr. Ding's laboratory and now mine is essentially that all GU pairs have important roles in PSTVd trafficking and replication, sometimes both. RNA loops also are critical for RNA trafficking and or replication. And surprisingly, different GU pairs and 3D loops mediate PSTVd transport between different cell types, and very often in a directional manner. So that suggests that all plasmodesmata are not the same. That is, there are different requirements for RNA to move between different cell types. So just to amplify that point, this is a diagram of a cross-section of a leaf. In this case, it's an inoculated leaf. We typically inoculate on the epidermis, which is the upper grayish cell layer there that you see. And the viroid needs to move out of that layer down into mesophyll cells below. Of course, it can move laterally within layers as well. And then it gets into the phloem, shown by the red arrow, and for long distance transport. Now, up and to the right, you'll see a systemic leaf, and you'll see the viroid exiting, the RNA exiting the phloem, and then subsequently invading that leaf as well. Excuse me. Now, as an example of directional transport, the PSTVd loop 27, which in the PSTVd genome shown at the top is the terminal loop there on the right, we found that that particular loop, an intact loop 27, is required for the PSTVd RNA to exit the epidermis in the inoculated leaf and invade cells below. Surprisingly, if we put the viroid on the other side of the barrier, that is, the mutant virus on the other side of the barrier, for example, by inoculating it into the phloem, we find that it's perfectly capable of moving between all other cell types and even invading the epidermis in systemically infected leads. So it's essentially a one-way ticket required for epidermal exit, but not entry. Fascinating. So there's a diagram here of a very crude one of plasmodesma, of a plasmodesmata, if you will. And we know very little about how these things function. In fact, there are proteins there that regulate the gating, and presumably these are different between different cell types, but we really know very little about how this occurs. And really the next frontier will be to figure out what host factors recognize the elements that are required for this RNA transport. Okay, now back to PSTVd replication. PSTVd replicates in the nucleus where it hijacks host DNA-dependent RNA polymerase II. This is the enzyme that transcribes DNA into mRNAs. It transcribes PSTVd RNA using a rolling circle mechanism, as we'll see in a minute. We don't know much about how it manages to use a non-native template RNA in this case, but we do know that a plant-specific variant of transcription factor 3A enhances Pol II transcription of PSTVd. Now, 3A, TF3A, is actually a factor that typically works with RNA polymerase III. So there's a lot we don't know about this process either. The only other thing that I can tell you for certain is that some of the structured loops and GU pairs are critical for PSTVd replication. The only other example I'm aware of Pol II transcribing an RNA is hepatitis delta virus. Excuse me here. Okay. Now, I want to bring your attention to a couple of things. First, on the far left, the PSTVd transcription domain. This is a region where Pol II binds and TF3A, CF, 7ZF, the transcription factor, bind within that domain. So it's a region that's critical for replication. Another region is in the center, the central region, where you see right around between loops 13 to 15. This region is also essential for replication as well as for processing the genomic RNA, as we'll see in a minute. Now, loop E is a motif that was initially recognized in ribosomal RNA. So this is a motif that's found in a lot of different RNAs. And you can see there's an annotation there of the non-canonical base pairing within it. I'm not going to have time to go through those non-canonical base pairs, but just be aware of that. Now, something I will come back to in this talk are the terminal conserved region and the central conserved region. And I've indicated those with red bars on the genome sequence. We'll come back to these in a minute. These are obviously conserved because of their critical roles in replication and genome processing. Now let's drill down on the rolling circle replication of PSTVd RNA. In the upper left, you'll see a circular plus strand representation of the genome. Okay, this is by far the most abundant RNA in infected cells. It's the infectious form of the viroid. This is transcribed in the nucleus by Pol II and in a rolling circle mechanism which produces genome concatenamers. Up to at least 7 of these minus strand concatenamers are generated in the nucleus. And the minus strand concatenamers subsequently serve as template for plus strand concatenamers in a second Pol II transcription event. Now, these plus strand concatenamers are then processed to monomers by a host ribonuclease and circularized by host DNA ligase. So the goal of this study was to assess Pol II error rate when transcribing PSTVd RNA, which of course is a non-native template. And we wanted to do this in the absence of selection. And we reasoned that the transcription errors that might be produced during production of minus strand concatenamers are not subject to selection. Therefore, this might be a way to get at our, to accomplish our goal. Okay, so why care about viroid and virus mutation rates? Well, I think we all heard a lot about coronavirus mutation lately, so it shouldn't be a surprise. Accurate calculation of mutation rates is critical for evolutionary studies, of course, but even more importantly, to evaluate adaptation potential. and we're all very concerned with that. So estimation of in vivo mutation rates is a tricky business, and it's complicated by selection, which of course favors some mutations and eliminates others. Now, one way investigators have got around this is by examining lethal mutations, because these are unlikely to be propagated, so their frequency should equal the mutation rate. This lethal mutation method has several limitations, probably most obvious being its inability to survey the entire genome. And we wish to do that, so we developed a simple method to estimate PST mutation rate based on the Pol II error rate across the entire genome, and again, in the absence of selection. Now, polymerase errors are the major source of mutation in virus and viroid genomes, and so they are key determinants of adaptation potential. So DNA viruses have high mutation rates, but relatively, considerably lower than cellular mutation rates. And these range between 10 to the minus 6 to 10 to the minus 8 substitutions per nucleotide per cell infection. This is one way to describe mutation rate. RNA viruses typically have higher mutation rates between 10 to the minus 4 to 10 to the minus 6 due to the error-prone nature of viral RNA polymerases, which do not proofread as DNA polymerases can. Coronaviruses, by the way, are an exception. They do proofread because they have very big genomes, and the mutation rate would be too high without a proofreading function. But still, mutation rate is still high. Now, using the lethal mutation method, there have been several estimates made for viroid mutation rates. Chrysanthemum chlorotic model viroid and eggplant latent viroid, for example, have been assessed using the lethal mutation method. And reports have shown anywhere from 1 in 4 mutation rates ranging from 1 in 400 to 1 in 1,000 nucleotides. These viroids are of the absent viroidae, and they replicate in the chloroplast using chloroplast RNA polymerases. Previous estimates for PSTVd have ranged from 1 in 100 to 1 in 7,000 nucleotides. In other words, around 10 to the minus 4, one mistake, in 10 to the minus 4 nucleotides, suggesting that Pol II transcribes viroid RNA more accurately than the chloroplast enzyme does. But again, these estimates were made using the lethal mutation method, and we wish to assess the Pol II error rate and the PSTVd mutation rate across the entire genome in the absence of selection. So this is our simple strategy for doing that. In panel A, you'll see a circular plus strand genome with a red dot. That red dot indicates a mutation. So this is a pre-existing mutation in a viroid genome. It's presumably a variant, and it's transcribed by Pol II in the first instance to produce a minus strand dimer. So that mutation should be present in each of the monomers. In other words, it should be shared by each of the monomers, again, indicated by the red dots. However, a mutation generated during that transcription process will not be shared by other monomers. That's the yellow dot. And we refer to those as unshared de novo mutations. Those are generated by Pol II during that transcription event. And so our strategy was to sequence minus strand dimers and look for unshared mutations. And the protocol is pretty straightforward. Extracts from Nicotiana benthamiana plants, this is a model tobacco that we use as a host, were infected with PSTVd. cDNA was prepared from the minus strand concatemers using a reverse transcriptase with a low error rate. and dimers were amplified from that cDNA by PCR using a very low error rate polymerase for that purpose. And we excise the dimers from the gel, which is shown in the middle, and subjected them to Sanger sequencing to avoid mutations generated during deep sequencing. We did this in 3 independent replicates indicated RA, RB, RC. Now, this is a lot of numbers on this table, but I'll talk you through it, sort of summarize things. Each of these replicates had similar numbers of dimers sequenced, including a control on the right with the red asterisk. So in total, in the red boxes, we sequenced a total of 261 dimers, which is more than 500 genome equivalents, and more than 187,000 total nucleotides. And in those 187,000 nucleotides, we found 102 unshared de novo mutations. So from that, we could calculate an error rate of 1 in 1837 nucleotides. By contrast, we found only 1 shared mutation in all of these experiments. And the control, which we created starting with a cloned PSTVd dimer we transcribed in vitro with T3 RNA polymerase, we found only 1 mutation. So the background mutation rate was negligible. Now, I'm going to go in a little more detail into the Pol II error rate and mutation rate in the next slide. So again, we saw an apparent Pol II error rate, an observed error rate, if you will, of 1 in 1837 nucleotides per transcription cycle, or to put it another way, an error rate of 5.4 times 10 to the minus 4. From this, we extrapolated a mutation rate because we couldn't look at it directly. We extrapolated based on 2 transcription cycles and assuming the same error rate for each. Again, assumptions, but the best we could do. To put it another way, twice the mutations in the same number of nucleotides sequenced. And we came up with an estimated extrapolated mutation rate of about 1 in 10 to the minus 3, which turns out to be very similar to the chloroplast replicating viroids. Now, to think about the Pol II error rate again, our initial question, we're using a non-native template here. What's the Pol II error rate for mRNA transcription? And the best estimates that I could find were from yeast and C. elegans, where they agreed, and both were about 4 times 10 to the minus 6 per base pair. So comparing the Pol II error rate and assuming similar transcription machinery in Nicotiana benthamiana, we can say that Pol II transcribes PSTVd RNA with much lower fidelity than DNA templates, about 100-fold lower fidelity. Nevertheless, these are some of the unshared de novo mutations. I got ahead of myself there, that we found, and you'll see they're listed for all 3 replicates. And again, too many numbers to look at, but about 30% of the dimers sequenced actually contained an unshared mutation. Most of them had only 1. Some had 2 or 3. And all but 1 involved a single base substitution. Now, some mutations appeared in multiple genomes. It turns out only 64 of those 102 mutations were actually unique. Some appeared in multiple genomes and in many replicates, in more than 1 replicate. And mutations observed in more than 1 replicate are shown in red. Okay. And some of them were found in all 3 replicates. Despite the vast difference in fidelity on different kinds of templates, the Pol II mutation spectrum on DNA and PSTVd RNA templates was actually quite similar. On the left, you'll see transitions were much, much more likely compared to transversions by way of base substitutions. Base substitutions were the most common type of mutation. They accounted for about half of them. Single base insertions and single base deletions each accounted for about a quarter of the mutations, and the majority of them occurred at homonucleotide tracks, as illustrated by the examples here of A123A, 124, which is an insertion between 123 and 124, and delta 180U, which occurs in a run of U residues around position 180. So on DNA and RNA templates, Pol II is more likely to commit transition errors versus transversion errors, and preferentially commits indel errors at homonucleotide tracks. I think I'm going to skip this slide here for the interest of time. What you're looking at is a diagram of the genome again, indicating the mutations that we found. Now, to the outside of the genome are mutations, are the de novo mutations, and inside are mutations found in progeny populations. I'll talk about that in a minute. For the moment, let's just talk about the ones on the outside of the genome sequence. So the blue arrows are base substitutions, the red arrows indicate insertion events, and the brown triangles indicate deletions. Now, the arrows indicate the most common de novo mutations we found. A123, 124, we actually found 19 times, and delta 180U, we actually found 6 times. So what you'll notice is that the mutations appear to be clustered in certain regions of the genome, but what really jumped out at us was that mutations were mostly absent from the central conserved region and the terminal conserved region. In fact, if we separately calculate the mutation rate for those regions compared to the remainder of the genome, we find that those two conserved regions are transcribed with an error rate about 4-fold lower than the remainder of the genome. So the central conserved region and the terminal conserved region sequence conservation is a consequence of both function-based selection and selection for greater Pol II fidelity. Now, in a previous study, we deep sequenced progeny from PSTVd infected Nicotiana benthamiana plants, and I won't go through this in great detail, but we looked for mutation hotspots that were found in those variants above a background threshold of 30 reads per million. And we identified hotspots that were present in 10 unique sequences per million reads above that threshold. And when we compared those, and those are shown on the inside of the genome, and when we compared those with the de novo mutations, it was clear that most of the de novo mutations were lost through selection. Not surprisingly. This table just lists the 24 mutation hotspots we identified, and the ones in red were the ones that were also found in our dimer sequencing, that is de novo mutations. So most of them, most of the de novo mutations are either eliminated or negligibly present in the progeny populations. So, to summarize, the Pol II error rate we found transcribing PSTVd RNA is about 100-fold greater than error frequencies observed in mRNA populations. Nevertheless, Pol II error spectrum is similar on DNA and RNA templates. The PSTVd mutation rate that we calculated, or extrapolated, I should say, is very high, about 1 in 1,000 nucleotides, which is approaching 1 mutation per 3 genomes generated, which is similar to the chloroplast replicating viroids. And compared with progeny population that we studied by deep sequencing, most of those de novo mutations are eliminated or minimized by selection. Surprisingly and remarkably, I think, the Pol II error rate in replication critical regions is 4 times less, 4-fold less than the remainder of the genome, suggesting those regions are selected both for function and enhanced transcription fidelity. And this biased Pol II fidelity suggests a novel strategy to promote replication while allowing abundant sampling of sequence space in the remainder of the genome. And frankly, I would expect that perhaps other viruses, in addition to the viroids, have developed such strategies as well. So, that concludes the talk, and I just want to acknowledge once again John Wu in red there for essentially doing all of the work that I talked to you about today. And thank you for listening, and thank you for your time. Thanks very much.